I'm going to tell you a little bit about the work I've been doing. I've met some old friends here tonight, so some of you may have heard some of my talk, my speech in the past. But hopefully you'll pick something new up out of it. The slides are busy and complicated, and the topic is very complicated. But hopefully I can try and extract something out of that, something simple that you can take away from it. So I'm going to talk about the revolution in our molecular understanding of what Parkinson's disease is. And it's not a revolution in diagnosis. The genetic work that we're doing, that I set out to do, and I think that most of the field is doing too, is not with the primary goal of predicting who will get Parkinson's disease. That's not the purpose for me. It's really trying to understand what are the components from a molecular point of view, from a cellular point of view, that are perturbed by Parkinson's disease. And it's taking extreme examples, families with many people affected, multi-incident kindreds we call them, families with many people affected with Parkinson's disease, and taking these extreme examples and mapping the genetic defect that they have, which we can attribute to their disease. Taking these extreme examples then and then putting these pieces of information together gives us a view of what's going on in the cell and in the body. And that view allows us to think of creative ways, novel ways to intervene and to slow the progression of the disease, to halt the disease in its tracks. That's the ambition, and I think we're well on course, and you'll see what I mean. To start off with, I remember showing this slide once before, and I was surprised at the audience's reception to it, because many people think Parkinson's disease is one entity, and it's not. It's a really heterogeneous disorder that's lumped together under one Gestalt diagnosis. And for instance, here on the slide, there are many forms of Parkinsonism that are listed. The one that we all know probably best is late onset, typical Lewy body Parkinson's disease. But there are many, many different forms of this. And the genetics helps, I think, to explain some of this heterogeneity. Because in different parts of the world, Parkinson's disease, late onset, typical Lewy body Parkinson's disease, has a different etiology too. There are different genetic risk factors in different parts of the world. And I'm going to show you some of those tonight. At the moment, we're doing we're doing many studies, but I've got two new studies that I'm really busy with. One is mapping the excess incidence of Parkinson's disease in the Faroe Islands, which is a group of islands just north of Scotland. And every patient on the Faroe Islands has contributed a DNA sample to this study, including 40 that have passed away and have given their brains for a post-mortem. So it's 160 patients in total. And we're using this collection of individuals, their DNA samples, their clinical data, to map their genetic etiology. The Faroes has 3 times the incidence of Parkinson's disease than it should. So I think we should be able to pin down what's driving that. We're also mapping the Parkinson's disease, the Parkinson's-Dementia complex of Guam, another small island in the South Pacific. And that work's going ahead very well too. And I'm not going to talk about those today, but next time, hopefully I can give you some new genetic insights from those. But there are many forms of Parkinson's disease. But fundamentally, they're all attributed to the loss of dopaminergic neurons. And here's a dopaminergic neuron for you. It's complicated. and to try and tell you otherwise would be crazy. But this is just an illustration of a dopaminergic neuron. And what you can see here, I think I have a pointer, so I don't know if the pointer comes. Oh, it doesn't matter. What you can see here is the main part of the cell is the cell body. Of the neuron has a cell body. It has lots of dendrites emanating from it, and it has one long fiber, the axon, which terminates in these synaptic connections. These, if I had well, I couldn't point at it anyway, but the end of the axon has a lot of tiny synapses, synaptic connections, which basically boutons, which will abut to other neurons, right? Connect to other neurons and communicate, allows the cell from this neuron to communicate to another neuron. Each dopaminergic neuron has something in the order of, although this is illustrated here with an axon fiber leading up and with a few presynaptic connections there, each dopaminergic neuron has something in the order of about 450,000 of these innervating the striatum. It's a very complicated beast. Right, to try and these cells are lost. Each of us has about 700,000 of these neurons in the substantia nigra, in the black substance of the brain. But as we age, they die. And in Parkinson's disease, they die. And by the time somebody manifests with symptoms of Parkinson's disease, they've lost a good half of them, if not more, of those dopaminergic neurons. They've gone. They're no longer there. So trying to study this disease is a challenge, right? Because the tissue that you actually want to study, not only is it buried deep within the midbrain, but it also tends to be ablated or dysfunctional in those patients who have the disease. And to remove it would be disastrous, impossible. So how do you get to it? Well, we've been taking the view that a genetic approach, and I'm going to explain what that genetic approach is in the rest of the talk, but it really the synthesis here and some of the things that hopefully may resonate with you now or in the future are these three key themes. What's shown here in this slide is a synapse. It's basically the connection of one neuron to another. And inside the synapse are all these little round circles. They're vesicles. They contain neurotransmitter, in this case, dopamine, which is released in the striatum. But they also contain many other structures. And basically, this part of the cell is responsible for producing the neurotransmitter, releasing it into the extra cellular space there, into the synaptic cleft, and then re-internalizing that membrane that's fused with the outer plasma lemma of the cell, of the presynaptic terminal, recycling that membrane, pinching it in again, invaginating it again, and then recycling it. There are a number of processes here at play, and really the genetics points to three of them. They won't mean much, and to explain them to you in non-technical terms will take hours. But there are autophagy, which is basically a process whereby the cell traffics and degrades proteins it no longer wants. They involve mitochondria, which mitochondria produce the energy of the cell. This is a very energetic process. It takes a lot of ATP. And central to this, of course, is synaptic vesicle endo and exocytosis, so the release and reuptake of the membranous vesicle. So it's a very active process. And these three things, this is the, to my mind, the holy trinity, if you like, of Parkinson's disease. And I'll tell you why. Because it comes from genetics. My old mentor used to say to me, genetics is truth and everything else is opinion. And basically, he's right. He's right, and the reason he's right is because most of the genetic studies we do rely on information, clinical information from families with the disease, in which many people in the family are affected. It doesn't rely on any preconceived notion about what might be the cause, or what the gene might be, or what the protein might be in the body. It's basically a very unbiased way of trying to identify a component. And from this work over the last 15 years now, we've identified 5 major genes with mutations that lead to late-onset Parkinson's disease. They're listed on the slide in bold. Synuclein, LOC2, EIF4G1, BPS35, and DNAJC13. Strange-sounding names. And what does it mean? There's a lot of other genes there for Parkinsonism and atypical Parkinsonism. And I think there's a synthesis coming whereby all of these genes can work together. The proteins that they encode work together to basically lead to the same overt condition at the end of the day. And I'll get into a bit of that synthesis towards the end. But these genes on this slide, basically, if you have a mutation in these genes, these mutations track down the family line. So 50% of each generation is affected, and 50% of their offspring are affected. But of course, you still have to get to, it's late onset typical disease, you still have to get to 60, 70 years of age to manifest the symptoms, right? So age is still the biggest risk factor for the disease. There are many other things that in your life that may occur that will either slow down or stop the symptom progression, stop you manifesting the disease at all, or you may never reach that age. Who knows? So, so it's not your destiny if you have a mutation in these genes to get the disease. Okay, but nevertheless, they do track down the family line. They are linked to disease. This list of genes on the next slide, some of them overlap. These genes are what we call susceptibility genes. They've come through a different approach. These genetic risk factors don't segregate down the family line, but they are found at increased frequency in patients with Parkinson's disease versus age-matched, ethnically matched, gender-matched, whatever, control subjects. Their contribution to risk is far less. So typically, at best, they double the risk of disease. But nevertheless, they're also very important and influential in terms of our understanding of what the disease is. There are at least now 20 genes for Parkinson's disease, some of which, I said, track down the family line, some of which are just susceptibility. Synuclein is the first one, and we just recently found a new mutation in synuclein H50Q. These mutations, these point mutations, are described here on this slide. A contribution to this was identifying multiplication mutations. And it was with the help of this family, this gift from this family, who I first met in Seattle, actually, in 1998, that we were able to pin down that multiplications, genomic multiplications of alpha-synuclein were enough to cause the disease down the family line. The lady in the middle holding the baby has Parkinson's disease, relatively young onset Parkinson's disease, with dystonia and a masked face and all the features of the condition. We went on to show that having more copies of alpha-synuclein basically is a bad thing. Normally, most of us have 2 copies of alpha-synuclein. But these patients with Parkinson's disease can have 3 copies or 4 copies of alpha-synuclein. And the more copies of alpha-synuclein you have, the younger your age at onset, and the more rapid your disease progression, and the more comorbidities you get, whether it's cognitive decline or autonomic problems, constipation, whatever. With 4 copies of the gene, so doubling of dose, 100% increase of dose, the mean age of onset is around 35. And the mean duration to death is about 10 years. But with 3 copies, so that's just 1 extra copy, the mean age of onset is around 70. And there's quite a window. So half the people out there with 3 copies of alpha-synuclein gene who are 70 years of age haven't yet manifested the disease. Even though it appears to segregate down the family line, there's a lot of people out there who are elderly who haven't manifested the disease and probably won't in their lifetime. So it's just a small increase, We found that not only do we see this in families where you have multiplications of alpha-synuclein contributing to disease, but common garden variety variability in the synuclein gene also contributes to disease the world over. So every population that's ever been studied now, and these studies have engaged more than 30,000 patients with Parkinson's disease around the world. Every study that's been done shows that alpha-synuclein is a risk factor for Parkinson's disease in most patients, although it contributes only a small proportion of disease risk. For most patients with idiopathic Parkinson's disease, we don't actually understand what the underlying variability is or what it tags. We're going to find that out, I think, in the near future. However, synuclein changed the field when it was discovered in 1997 and with multiplications in 2003, and understanding this mechanism of increased dose that leads to Parkinson's disease and leads to invariably to Lewy body disease. Antibodies to alpha-synuclein were discovered to bind to Lewy bodies, the hallmark of Parkinson's disease in the brain. And so this is a slide of Lewy body pathology in the brain of a patient with Parkinson's disease. There are 4 sections there, different regions of the brain, but the nigra is included. There's pigmented neurons. And this is what you normally used to see on H&E staining in 1997. And in 1998, this is what you saw with synuclein antibodies. All of this brown pathology, all this occult pathology is abnormal. It shouldn't be there. It's all alpha-synuclein staining. So although alpha-synuclein was found in rare families, and there's only a few of these families in the world, everybody with Parkinson's disease, pretty much with garden variety Parkinson's disease, has this pathology in the brain. We realized by studying many people who've come to autopsy and looking at disease duration and the burden of alpha-synuclein immunoreactivity in the striatum, that the amount of synuclein basically correlates with disease duration. So it seems to be a progressive phenomenon. And Heiko Brack recently was nominated for a very large prize because of his work. He's an anatomist. He's been studying the spread of synuclein in the body and in the brain. And he's shown that you not just get synuclein in the brainstem, in the midbrain, but you also find it in the gut, in the myenteric plexus, in the gut nervous system. And it may ascend, it appears to ascend, perhaps via the vagal nerve to the brainstem, to the midbrain, and then into the limbic system and into the cortex. And this is a very interesting and curious observation, because Parkinson's patients get constipation as an early feature, long, long, long before any signs of movement disorder. In terms of synuclein's normal function, realizing this was a gene for Parkinson's disease, it's very easy once you've got this tool to play with it, to knock it out, to overexpress it, to make models of the disease condition. And this is really the path to therapeutics. So knocking out synuclein and all of its homologs basically causes neurodegeneration in mice. It's absolutely required to have at least some of synuclein or synuclein homologs. And it was shown that synuclein's got a very active role in synaptic vesicle exocytosis. Not going to go into it too much. It would take too long, but you do need some alpha-synuclein. More recently, it's been shown that if you take synuclein fibrils and inoculate them into a mouse brain, or actually even into a primate brain that is not published yet, this synuclein pathology can spread from one cell to another. And this was shown early on. I think the best illustration probably is from fetal grafts that were used for some Parkinson's patients. They subsequently show from autopsies that the fetal grafts actually developed Lewy body pathology, and they acquired the synuclein of the host. This has now been shown to be a phenomenon, this permissive templating in the brain. And perhaps it's a permissive templating that starts in the gut and ascends via the vagal nerve and gets into the brain, or goes via the olfactory bulb. We're not exactly sure, but it seems to be the current theory, and it seems to be standing up. And it seems to be associated with progression of the disease. So here's the good news. We know what it is. We've got a fair idea where it starts. And we realize what the mechanism is, simple overexpression. Right? We also realize it's an equilibrium. If you knock it down too much, you have a problem. But if you knock it down just a bit, maybe you've got a therapeutic. So we set up on this road some years ago, and we focused on making what's known as, using a technique known as RNA interference, to basically stop alpha-synuclein from being expressed. We can't stop it absolutely, but we're pretty good at hampering it. And so we've carried out these pilot experiments in mice, and you can see a mouse here treated on one side of the brain, and basically we could measure, in this case it's the hippocampus, looking at synuclein expression. You can show that we can knock down synuclein expression in the mice by about 50%. The effect lasts about 2 or 3 weeks. And that's a pretty good effect for a drug with a washout. We also went on to show this in non-human primates, and show that this approach was reduced the toxicity of other toxin treatments in squirrel monkeys, and was efficacious to do that. So it's just one approach. Others in the field have been using passive immunization approaches. And there are several companies now, Biogen, IDEC, Elan. It's no secret, working on these types of approaches, Alnylam, ISIS, I should name them all probably. I don't know about Abbott. It's no secret in the field that many of these big pharma companies now have multi-million-dollar research investments to develop these types of therapeutics. This one here is work that was published by Eliza Maslier last year in 2011. Which shows that using a passive immune approach, where basically they gave these mice intraperitoneal injections of an antibody to alpha-synuclein, they could rescue the mice that were developing, otherwise we were developing synucleinopathy in a form of Parkinsonism. It's probably most elegantly shown here in this slide, where I think if you look at the third column, these are the synuclein mice that This is a water maze test, a behavioral test, whereby the mice are swimming in milky water. And normally they swim to a platform and they sit on the platform and they try and get there as fast as possible because they don't really like swimming that much, right? And, and basically you train them over a period of days. Mouse that are transgenic for alpha-synuclein can't do this task very well. They're suffering from neurodegeneration. But if you use the passive immunity approach with this antibody, which is the last column, you can pretty much rescue these animals. And so the hope is that this may be an approach that could be tried with patients, probably with synuclein multiplication patients first, in the near future, to basically slow down and prevent this, whether it's permissive templating or whatever, whether it's a passive spread of synuclein in the body or brain, but to slow down and hamper or prevent this process from occurring. And that's great news because it would help with the problems with constipation, potentially, if it was given orally, and it would help if it can get across the blood-brain barrier. And there's a lot of recent success with these types of approaches with a number of companies, that it can actually perhaps help with the cognitive issues that develop in Parkinson's disease. So it's phenomenal. It's phenomenal to go from a molecular clue and insight to a therapeutic, and we're talking 15 years. That's the typical kind of timeline. Moving on to LRRK2. This is the other major risk factor that Ted mentioned. It is a player in garden variety Parkinson's disease. This is a study that involved 15,000 subjects that we published in The Lancet last year. And it shows that not only is there common variability in LRRK2 that contributes towards risk in patients with Parkinson's disease the world over, because this study was done in Australia, Belgium, France, Germany, Greece, Ireland, Italy. You can read the slide perhaps. But it also shows that there's variability in the gene which is protective, which is responsible for successful aging of the basal ganglia. Probably the most common risk factor in leucine-rich repeat kinase 2 is this one, which we think is descended from Huangdi, the Yellow Emperor, because it originated about 4,800 years ago. And it's a G2385R variant that is found in 8% of patients with Parkinson's disease in Asia. So that's patients in Japan, that's patients in Singapore, patients in Taiwan, patients in mainland China. This is a huge market, pharmaceutical companies. Huge market, because you could think about making a drug that's efficacious for brain health, right? Not just for Parkinson's patients the world over. But our discovery for this was made in part with this fellow here, Jan Asli. When I was fishing with him in central Norway in the mid-2000s. And we initially had discovered a gene, leucine-rich repeat kinase 2, and Jan wasn't involved in that, but working with him, we had a number of families, 13 families around the coastline of central Norway, and we found that all of these families had something in common. This sequence, DYG, was mutated. Instead of being DYG, it was an S. It's a G2019S change. It's a single nucleotide change, actually. It's a 6055G to an A change, a single base out of 6 billion that we've inherited, that these guys have inherited. They had a different nucleotide at that position, which meant that they have Parkinson's disease that travels down the family line, these 13 families. Now, they're all related, and it was no surprise to me that they were all related because they're all in this part of the world. So, but what was obvious from looking at the sequence of this, there were a number of things that became very clear very quickly. This DYG sequence is the beginning of a hinge of a kinase. It's known as the activation segment of kinases. It's very well known in enzymology that these sequences start with DYG and they end in APE. And the hinge in this particular kinase is broken. And this is an illustration of it that I made back in 2005, just to show you there where the position of the mutation is. The hinge is that bit in yellow there with the G2019S beside it. And the ribbon of protein that emanates from it that kind of blocks this pore, this active site in the middle, is in purple. And this is always slightly ajar in G2019S Parkinson's disease, allowing, we think, more substrate access to the enzyme to be phosphorylated. And so the enzyme is slightly more active than it should be. And that's enough. But it leads to the thought immediately that if you can get a competitive inhibitor, so it's an inhibitor that doesn't work too well, it doesn't bind absolutely, but is reversible, to this specific protein, you can fix the problem. You know? It quickly became apparent from studies, from other group studies, both in Ashkenazi Jews and in North African Arabs, that LRRK2 G2019S was prevalent around the world. It turns out that 30%, so 1 in 3 patients in North Africa, and this is a huge, huge social and public healthcare burden, 1 in 3 patients with Parkinson's disease can be attributed to G2019S in North Africa, in Berbers. And we think that the spread of this was, and I gave a talk about this fairly recently, was spread by the Phoenician diaspora through this part of the Mediterranean basin around 3,000 years ago. Isn't it amazing that genetics can tell you so much? It's just wonderful, I think, about population history, let alone about enzymology. We're working with 258 families in Tunisia right now, and they very kindly, and I have to attribute this lecture to them, really, to all the families and all the individuals who've taken part in our studies over the years. They've very kindly given up their DNA, their time, their clinical information to be part of these studies, because we're working on a natural history study. These families obviously have individuals who are carriers who have yet to develop the disease, and they have people who are affected, of course. We want to understand where the disease starts and how it progresses, and what's responsible for that phenoconversion when somebody basically feels normal to all of a sudden walking into their physician's office and saying, I'm feeling a little slow, a little stiff today. What's that trigger that leads to that phenoconversion? We want to find biomarkers of this process. We want to, is there something in urine or stool or fibroblasts or sweat or saliva which also tracks with disease progression? Because ultimately we want to give these guys that kinase inhibitor to slow down the disease course. And we have to be able to have a short time window in which we can do this. 2 years, I think. Otherwise, the trial would not go forward. It would cost too much money. So we need biomarkers. There are some good ones. Imaging. It's part of the reason I came to UBC. It's great for imaging. We've been doing some studies of penetrance. This study here is, the curve here is represents the age-associated, actually, cumulative incidence. So it's the age at which individuals with idiopathic Parkinson's disease manifest symptoms. And each step on this is one or more persons in this study. This is LRRK2 Parkinsonism. And what's different about it is the women. I'm going into some science here, but it's kind of interesting. The women with LRRK2 G2019S get Parkinson's disease about 5 to 7 years younger than the men. The men look identical to idiopathic Parkinson's disease. We've found very, very little to discriminate them. A little less REM sleep behavior disorder, a little less in the way of constipation, but otherwise they're the same. But the women are different, and we don't know why. And it's not a cultural thing. We've made mice models, and several people have made mouse models. The fascinating thing about the mice models is they have dopaminergic deficit. So like human patients with the Parkinson's disease or with LRRK2 Parkinsonism, they have a loss of dopaminergic innervation in the striatum. They also have pathology in the brain, but it's not synuclein, it's tau. But then they're mice. And some patients with LRRK2 Parkinsonism also have tauopathy. I won't go into that unless someone's that interested. But our mice were overexpressing models, but we've also made knock-in models. We've also knocked out the gene, and we have a whole variety of these animal models. So I shouldn't go into that really, because there's a lot of animal rights activism in BC, but I think it's a necessary evil to develop therapeutics. But the knock-in mice models also have dopaminergic deficits, and they also have, and the knockout models, actually, they don't seem to have much in the way of deficit. They do have some synaptic transmission deficits, I'll show you in a minute. But interestingly, they have a kidney phenotype. And you might think, well, what's he going off about? This is tangential to Parkinson's disease, kidneys. But I'm interested in biomarkers, and I'm thinking if LRRK2 is playing a role in kidney, then maybe urine is going to be a great biomarker in urine, and we just have to look at that, first in the mice, compare the overexpressors with the knock-ins, with the knockouts, and then see what we find, and then look in the humans, see if we can find a biomarker. LRRK2 affects neurite outgrowth, which we saw very early, which is a bizarre thing, really, because LRRK2 is barely expressed in neurons at this time of development. But if you take neurons from the brain, from these animals, very early on in life, and put them in a dish and let them grow, you find that the neurons that have a mutation in LRRK2 basically don't grow as well, they don't arborize as well as their wild-type normal glutamate controls. Similarly, if you knock out LRRK2, the neurons grow excessively well. Too much, they branch a lot. And it happens at a micro level. So going back to that synapse I showed you right at the beginning, here's a picture here of synapses, of spines actually, little mushroom-shaped spines. It's in the middle panel on the left-hand side there. That's what you normally see in a non-transgenic animal, those little mushroom-shaped spines all the way along the neurite. But in a mutated mouse, you see far less of those in culture. So we're using these types of phenotypes in the animals now and applying drugs to them, LRRK2 kinase inhibitors, to see what effect we can have to ameliorate these phenotypes. I know it's not Parkinson's disease, and it may have nothing to do with it, but a whole series of different types of these combinations of these phenotypes can be now tested with these LRRK2 kinase inhibitors to see which ones are potent and which ones are having an effect. And they can be given to the animals in vivo as well to correct their movement disorder, their behavioral disorders, as well as their pathology. And these studies are ongoing at the moment. I think there's probably about $250, $300 million been invested in LRRK2 biology by Big Pharma over the last 4 years. So that's good news. This was a barren landscape. There was absolutely no investment being made in any of this in the last decade, till the genetics led the way. We've been doing studies of synaptic transmission, looking at the potential of these neurons, the electrical potentials of these neurons in knockouts and overexpressing animals, and we find some very interesting effects, very subtle effects, but that's what you'd expect in a late onset disease. The effects are subtle and they're chronic, and you need to have very exquisite, very sensitive measures to look at them. One of the most recent papers on LRRK2, which I found quite exciting, is actually work from flies that shows that LRRK2 has a role in synapses, in these connections between neurons, and it basically regulates the uncoating of synaptic vesicles that have been endocytosed from the membrane. There's a little schema there on the left-hand side, and LRRK2 has a role in regulating this flux. It's pretty remarkable. Whether it holds true of mammalian systems is another matter, but I think it well may. In the last bit of the talk, I'm going to tell you a little bit about the more recent advances. We are turning over the page 1 of Genesis right now in terms of next-generation sequencing. This is a huge revolution. All of the genes that were found in the last 10 years were found through very arduous, difficult studies, linkage studies that took years to do. A lot of work, a lot of effort. But now you can do it really almost in the blink of an eye with a next-gen massive parallel DNA sequencer. And I have one. And it's great. And so in the last year, I wanted to tell you about this. This is the approach we use. 1 in 7 families, 1 in 7 patients with Parkinson's disease looks like this. 1 in 7 people with Parkinson's disease has a first-degree relative with Parkinsonism. That's the statistic, and I think it holds true. It's from Mayo Clinic, so it must be true. Olmsted County, at least it's true of the 4 states around Mayo and Mayo itself, Minnesota. But it's consistent across studies across the world. So 1 in 7 people with Parkinson's disease have a first-degree relative. And so we're taking these types of families. Here you've got a brother and sister who are affected in one pedigree branch, and their cousin, a male cousin, in another branch. And basically what we're doing is sequencing their genomes. And we're comparing the sequence variants that they have in common that are rare otherwise in the general population. And you can see by looking at the slide, on average we find about 85,000 variants per person when we do just exome sequencing. But when you compare cousin pairs like this and you rule out common variability and focus just on missense and nonsense changes, and you look at very rare things that you don't observe in the general population, you get down to more meaningful numbers, something in the order of like 43, 44, 101. Ultimately, shared between those 3 individuals, just 20 variants in common. And one of those 20, perhaps, we can attribute their disease to. And that's the work we're doing at the moment. We've just done 100 of these exomes from families around the world. And we've got some pretty exciting results. One of these results was VPS35. This is from families from Switzerland, Yemen, 2 families from Yemen, the US, and and from Tunisia. There's a family there from Tunisia. But there's lots of Austrian and German families now with the same mutation. This illuminated a whole new pathway that's involved in membrane recycling biology known as the retromere. And this was just published last year. And we're focusing a lot of work now on those spines again, on those synapses, because VPS35 is mislocalized within the spines of these neurons, at least in culture in transfected cells. It's a very new kind of pathway, and no one's actually ever studied this pathway before in neurons. It's only ever been studied in big fat cells that sit on the bottom of a dish and they're easy to observe, but not neurons, so we're doing that. We found another gene in the same pathway very recently with the help of Mennonite families across Canada, and a big thank you to them because, again, it's a humanitarian gift that they've given us. They don't get anything out of it particularly, you know. And knowing about this gene mutation within the family, I don't think it's particularly useful or helpful to them. It's a late onset disease, like I said, and I don't think it's that predictive. We don't know what the window is of age of onset and stuff like that, and it's probably at least 20 years. So it's not your destiny. But nevertheless, they put aside their beliefs to contribute DNA samples, and we identified a new gene called DNA JC13, or receptor-mediated endocytosis 8, which has just been announced, which is a very exciting new discovery, and it fits very much in the same pathway as VPS35. In the literature, this one just came out as well this year, which is actually another component of that synaptic vesicle endocytosis mechanism. It's known as auxilin, or DNA JC6. And so it's a third gene, actually, that falls within the same pathway. Perhaps the fourth, if you consider LRRK2's function to be principally within synapse and principally involving endophyllin A. Put together, and you've got a schema like this. It's not really, it's challenging to put all the genes together or the proteins together in one pathway. And the reason why is because I think the relationship in part is temporal. It's not necessarily physical, and one isn't just necessarily upstream of another in a linear pathway, but there's a temporal relationship in these different constituents of the cell, and working together, I think, focused certainly within presynaptic terminals and maybe postsynaptic terminals as well. I think this is where they really come to play, and where subtle defects within them can lead to the loss of those terminals and dying back of axons, and ultimately the dying back and death of dopaminergic and other types of neurons that are lost in Parkinson's. So this is the schema here that's worked out in a cell, which is basically endocytosis. On the left-hand side, you've got the plasma lemma and the vesicles, the membrane, if you like, being reinvaginated and turned over, going into these bulk kind of endosomes, and then being trafficked through to autophagy for degradation, and along that process with a lot of recycling of those protein and membrane protein constituents being occurring. Okay, so that was a bit much, wasn't it? I thought I'd leave you. I thought I'd leave you with this. This is principally it, right? It's a bit. Genetics is a bit like, or the application of genetics is a bit like a mechanic trying to figure out how your car works, right? The first thing you need to know from a mechanic's point of view, if you're going to fix the problem and create a drug to do this, you need to know what the component parts are, right? And how they connect together to make that motor run, all right? And if you don't understand what the component parts are, and you don't understand how they fit together, and you have no idea how they're working synchronously in harmony together, you're never going to fix the problem. And you can ameliorate it with palliative therapies, levodopa being one of them. It's a superb drug. But you can't fix it. And you have to know what those components are to fix it. So that's what genetics is about, and that's what it means to me in this application. This is the lab, and I better stop now. I think I've probably run over time already. But the lab basically focuses very much with the Parkinson-specific research center at UBC, and with collaborators in 52 sites in 26 countries around the world, mostly national chairs of, or institutes of neurology, to do clinical studies, pathologic studies, and imaging studies. And we do a lot of population genetics, family-based genetics. All of those discoveries we make, we basically bang into mouse models, rodent models. Knock-in of the mutation, knockout of the gene, overexpression of the gene, And we create these model systems. We characterize them using very subtle methods, electrophysiology, for example, and primary cell culture. And then we work with pharmaceutical companies, moving up the other side, to think how treatments can affect that pathophysiology and correct it. And then we basically, having de-risked the enterprise, we try and persuade pharmaceutical companies to get invested. And I'm gratified to say that they are invested. And that's brilliant news for people with Parkinson's disease and people like me who work on it. This is the lab to be. So we're moving into the Center of Brain Health in the relatively near future, being 1 year's time. I came here with 2 people 2 years ago. 3, well, not including my wife and family, who's sitting here today. And with their support, the team has now grown to 30 people, and we're still expanding. And we're hoping to apply the same strategy, this strategy, to all of the neurologic disorders that are seen in the Center of Brain Health, or will be seen in the Center of Brain Health. So that's all of adult neurology. Because I think this is the same template, can be applied many, many times. And I think all of these age-associated diseases have actually a lot more in common with each other than differences. So we can learn from that. So, thanks very much, anyway, for your attention.
In one of the slides, you, I think, showed in your LERC or whatever, LERC2, that it sometimes had a protective effect and sometimes had a risk effect. Mm-hmm. What's up with that?
Oh, well, it's actually a wonderful finding. Yeah. I think it's LERC2 is a very big gene. 51 exons is a very big protein. It has a number of what we call domains that do different things. It's actually it's a whole pathway in one protein. It's a GTPase and kinase in one protein, and a lot of variability within that has now been found. At least 254 different amino acid changes in this protein. It turns out that some people have. We're not quite sure exactly why, but some people have a form of LERC2 which functions better than others. It's optimal, and it's basically a protective factor, if you like. It reduces your susceptibility to disorders of the basal ganglia, if anything. It's found at different frequencies, so that this variability, depending on the population, ranges from about 5 to 11% of different populations around the world, the protective effect. In terms of pathogenic effects, there are many, and many of them are population-specific. So, for instance, G2019S is found really, really high. 30% of all patients in Berbers in North Africa, 14% of Ashkenazi Jews with Parkinson's disease. Our R144G, R1441G, is another one that's found in Basques in the northern region of Spain. R1441C is found in Belgians, families in Belgium.
I was also intrigued by the sex difference. So you said, I think, that women were more likely to get it younger or something in at least one.
And that's new. We haven't published that yet. So this is something that we're working on at the moment, and I'm intrigued by it too. We've done the appropriate controls to think about. So this is the sex difference for LERC2 G2019S. We've done the appropriate controls in that we've looked at idiopathic Parkinson's disease and the incidence of that. And you don't see a gender difference between males and females in this population. So it isn't just that women are coming forward earlier, perhaps because of, they may see their physician more because of, children, or more comfortable with going to the doctor than men.
We go all the time.
Yeah. I didn't want to say that, but yeah. So, but there's something biologically different.
Yeah, I'm curious in terms, first of all, the onset time, because my understanding is that the actual diagnosis of the time, they're actually, person is actually about 80% into the disease, and so maybe that figure's wrong. But is there anything in your studies that have indicated that folk at-risk populations could engage in some diets, behaviors, exercise, etc., which might at least postpone the offsets? So if you know you're susceptible, like if your heart, you had rheumatic fever or something like that, you have a routine of being very fit, and maybe that prevents it.
Well, you're right. By the time an individual gets diagnosed with motor problems, with Parkinson's disease per se, they are already well in advance in terms of the disease's progression. In terms of the earliest symptoms and signs, James Parkinson's pointed out as constipation, and that's been recapitulated by the Honolulu Aging Study, which is this longitudinal prospective study of Japanese men in Honolulu. It followed them for many, many years, still following them, and has shown by looking back over those records over the years that people with, I think it was less than one bowel movement a day, basically are those more inclined likely to get Parkinson's disease. One example, REM sleep behavior disorder is another. There are many other manifestations of the disease prior to any movement or motor problems. So yes, you're quite right. In terms of genetic risk, you're quite right there too. For some of these genetic factors, sure enough, we can see signs of dopaminergic dysfunction, of the loss of dopaminergic dopamine release or reuptake in the striatum. So for instance, we've been engaged with families with LRRK2 Parkinsonism, and many of the carriers of that form of Parkinsonism have come forward. Young people have come forward for dopaminergic imaging at UBC, and sure enough, many of them are profoundly affected in terms of their imaging, but otherwise perfectly normal clinically, and will remain so probably. I think the youngest subject is 22, active ski boarder, it's a snowboarder, and his family has the disorder, and the mean age of onset in the family is in the mid-50s. So it'll be many years, but he is profoundly, affected. So there is obviously one of the things we would like to do is to get to these interventions, create these drugs, and then apply them as early as possible, ultimately. But the first pass will be to apply them to patients who are already diagnosed with the condition.
Certainly, it sounds like this is an amazing, advance by finding all these genes. Do you think that we've basically gone after most of the big genes, the ones that are explaining the largest proportion of patients, or if not, the other ones will be mainly involved with conferring a very, very small risk, additional risk, or not a large percentage of the population?
Well, the answer to that is I don't know, really, the honest answer to that. So a good example with GBA. So glucocerebrosidase mutations were found to be associated with Parkinson's disease. The odds ratio for those is about 5, so 5 times the increased risk if you're a carrier of one of these mutations. GBA is very common. The variability in it is very common. For instance, 5% of N370S is probably the most common variant. 5% of the Ashkenazi Jewish population actually have it, controls. Whether that equates to an excess incidence of Parkinsonism in Ashkenazi Jews due to GBA, we have yet to find out. But nonetheless, this wasn't picked up in GWAS studies, and it wasn't really picked up in rare variant studies either, because it's not really rare variants. And it was picked up by astute clinical observation. I think in countries, for instance, like the Faroe Islands, we're going to find another form of Parkinsonism, which will probably be very common to that particular population, and anywhere else that those Dutch settlers perhaps have emigrated to. And I think we'll find these pockets around the world. I think it's quite conceivable we'll find another LRRK2. And so we really don't know. We're not there yet. Some people, I read reviews all the time where people talk about having found 60% of the heritability of Parkinson's disease. This is nonsense. Heritability is something that it isn't bounded, right? We don't know how much it is until we've found all the genetic causation. So we can't say how far we are along the course, if you know what I mean. So I think there's a lot more work to be done. And I think we have the tools through next-gen sequencing to really go through it fast. But it's only with patient participation, basically, families. We have to get people engaged in this. And one of the things that bothers me a lot is this genetic discrimination, that for instance, in the States, it's illegal to discriminate against people who've taken part in, say, genetic research based on any findings. Just because they've taken part, it's illegal to discriminate against them. In Canada, there is no such law. And it would be nice to put something in place, because it's only with people coming forward, taking part, that we find these like pieces of the engine, if you know what I mean.